anyone understand what bacteria is when we talk about bacteria? Yeah. So they're different cells, basically. We've got cells in our body that have more of a structured form with nuclei and different things. I'm not clearly a chemist or a biologist, but bacteria don't have that really structured form.
Cool. So in terms of exudates for what we're talking about in the soil food web are one of the most important components of what's going on in this chart. So the plant, a healthy plant can release like 30 to 40% of its energy that it's making from the sun. It can release that in the form of exudates or liquid carbon into its rhizosphere, which feeds the bulk of this food web. There's some articles that talk about exudates being one of the main soil builders as well, from that energy that's getting put down there. Exudates are really important. Cool. Does anyone know what a fungi is? Yeah.
Me?
Well, it's a kingdom of single or multi-celled organisms that reproduce by spores. That's reproduce asexually. That include mushrooms, molds, yeast, and mildews. Fungi are heterotrophs, living by decomposing or absorbing organic material in which they grow. And they bridge life and death in the cycle of.
So the first trophic level in the soil food web, made by the Soil Food Web Institute, is your autotrophs, plants and organic matter. And our second trophic level is typically bacteria, fungi, and root-feeding nematodes. In the way that we've got this written out, we're going through bacteria, fungi, nematodes, and then protozoa. So it'll be a little bit back and forth in terms of the way that this runs, but I think it will make sense. So bacteria gets broken down into kind of four what's called functional groups. So all bacteria aren't serving the same function under the ground. We're going to break it down into decomposing bacteria, mutualistic bacteria, pathogenic bacteria, and lithotrophic bacteria. So the definitions we've got for decomposing bacteria. Does anyone have an idea of what the decomposers are doing?
Breaking up. Anything more?
So for all of them, we've got definitions on the paperwork. Basic definition of a decomposing bacteria is they consume simple carbon compounds, e.g. root exudates and fresh plant matter. Mutualistic bacteria. Does anyone want to read out the definition of a mutualist?
Forms a relationship in which both species benefit. For example, nitrogen fixing in legumes.
So, bacteria which make root nodules on legumes, which everyone I imagine has probably heard of. That's the type of bacteria that falls as a mutualist. And lichens, if anyone remembers the succession class, lichens are like the first to cover rocks and start to build soil in terms of the forest and ecosystem succession. Pathogenic bacteria.
They cause disease in plants by using cell wall degrading enzymes and toxins. Many prefer anaerobic conditions, e.g. the rod-shaped bacilli cell wall degrading enzymes and toxins.
It can get really dense really quickly, but we're going to try and keep it really broad. causes disease in plants is a really easy way to think about pathogens or pathogenic bacteria. Does anyone want to read out the definition for lithotrophs or lithotrophic bacteria?
Autotrophs able to produce their own nutrients through the oxidation of inorganic compounds. Example, nitrifying bacteria that uses ammonia and converts it into nitrite or uses nitrite to produce nitrate, or purple sulfur bacteria that transform sulfide into sulfur.
Does that make sense to you?
I'm really curious.
Yeah, no.
No, cool. This is come back
I
to
think
it.
oxidation of inorganic compounds.
Yeah, so this is, we can come back to mineralization and immobilization for the decomposers. The decomposers are immobilizing the root exudates and the fresh plant litter. The lithotrophs are mineralizing and immobilizing ammonia, nitrites, and nitrates, or sulfur from sulfide. Pathogenic bacteria fall under the heterotrophic, so they're going to be consuming living tissue on plants typically. And your mutualists, they're setting up a two-way street of exchange, mineralizing and immobilizing, usually forming associations with plants or other bacteria sometimes. Some people.
How else would you define lithotrophs without saying oxidizes inorganic compounds?
You can think about lithotrophs as an autotroph, so they're, in terms of producing energy they're a producer, creating energy for all of the consumers further up the food chain.
Is that like a free-living bacteria?
So cyanobacteria fall under lithotrophic bacteria, and they're one of the first bacteria that started on Earth and has created the environment, everything that we live in.
Can you think about it as in they sequester they use the gases in the air to make energy, which then they're eaten by, and that energy gets transformed into a new form into something else.
And as we get further up, we'll be moving into predators like our Protozoa and Nematodes, which are mineralizing all of those nutrients that are immobilized by our functional groups of bacteria.
Does that make sense? Yeah? Yep.
Cool. We've got to move on to fungi.
Any other bacterial questions? Yeah.
Pathogenic are always bad guys?
Not necessarily. So these functional groups, bacteria that fall into those functional groups, aren't always going to be in that group. And I've got some examples in the fungi to try and explain how things can jump through. They're not always a pathogenic bacteria might be beneficial to some plants, whereas it might be pathogenic to another plant. So something that falls under pathogenic might jump between mutualist and pathogenic. And vice versa. A mutualist might become pathogenic.
In regard to the mutualistic bacteria, do bees need more nitrogen than other plants?
Say that again.
Do legumes need more nitrogen than other plants since they form that relationship with the bacteria?
So my understanding of legumes and nitrogen fixing is because the plant's releasing exudates to feed that bacteria inside of those nodules, if the plant has access to a nitrogen source in the soil, it won't share any of its energy to the bacteria in the nodules because it's getting its nitrogen from somewhere else. So it only will fix and accumulate nitrogen if it's limited in the soil, is my understanding of that.
But what Em's saying is that do those plants need more nitrogen? Is that why they have that predominant exudate?
Yeah, so typically legumes are identified because they have big beans and big pods. Those pods take a lot of nitrogen to put together.
What's that? Protein.
So if you let things go to flower, they actually not
Providing nitrogen for the rest of it. Yep. Yeah, absolutely.
Yeah, the way that we manage legumes is really important to try to get that nitrogen available to another succession of plants. Letting them go to flower, they're going to use all that stored nitrogen. But then that nitrogen's in the seed pod. If you when that compost, that nitrogen's still in the food web.
Do you chop in and drop it before the flower?
Yeah, ideally. Yeah. I think we would have managed a lot of the legumes a couple of months ago, but I think the decision was made that we were going to let them go to seed and collect the pods to eat. So that's why we haven't done it this year. Fungi?
Does anyone want to redefine saprotrophs? Does anyone know what a saprotroph is? So decomposing bacteria are also saprotrophs. Does anyone want to read out the definition for a primary decomposer under fungi?
First to grow on a twig, grass, or wood chip, and typically fast-growing using enzymes, break down plant matter into varying stages of decomposition. For example, oysters and toxin mushrooms.
Does that make sense too, right? There's a lot more that fall into primary, but that's a couple of examples. Secondary decomposers.
works in concert with actinomyces, other bacteria, fungi, and yeast. They prefer environments that are more biologically complex than primary decomposers. Example would be button mushrooms and Staphoria.
So I chucked Staphoria in there because it does well in partially decomposed material, but like I believe Bunya has King Staphoria in their Permi patch at the farm, and they're feeding it like fresh wood chips. So that's an example of a fungus that can be a primary decomposer, but can also be a secondary decomposer. They're not quite as fast as your primary decomposers, so they might be present in fresh organic matter or fresh residues, but it might not be until that material is partially decomposed that they're going to start to fruit. And once that material is partially decomposed, there's going to be a much more complex biological environment happening in that material.
It's to do with what's there and speed, as well as it being like the actual ability to break down from like fresh wood chips.
And a lot of primary decomposers might not like it if they're not first on the scene, and there's a lot of bacteria and other biology going on.
I just recalled that Psilocybin cubensis is also considered a type of Staphoria, and it grows off of cow dung, so that would be an example of a different stage of breaking down, but then they can also grow off of like cocoa fiber, so there's kind of a gradient in where the type of material is.
Nothing's stuck in any of these boxes, but they're really useful words for describing where something might sit in terms of succession, where that organic material is on its way to being mineralized, or if it's still immobilized. And we've got tertiary decomposers. Does anybody want to read that one?
Termed soil dwellers, they come up in areas holding little decomposable material and rely heavily on highly complex microbial environments. Field mushroom and button mushroom also each.
So again, the field and button mushrooms can sometimes fall into that secondary decomposers as well. But they will do really well in environments that don't have a lot of decomposable organic matter, and they'll still have the ability to fruit when humidity and temperature let them fruit when the conditions are right. Parasitic fungi. Parasitic fungi are predators that endanger the host health. Most are microfungi. And examples are the honey mushroom again, oyster, and reishi. So I put those three examples. Oyster and reishi would typically, you would call them a primary decomposer, but they're like an opportunistic parasitic mushroom. So if there's a tree that's on its way out in a forest that's not getting the light requirements that it needs, or it doesn't have the nutrients that it needs, and that tree is dying, your oyster and reishi type mushrooms will actually attack that tree, become parasites, kill that tree, and be the first on the scene to decompose that material, which is where they want to be, but they will finish dying organisms off to get access to that organic matter.
Which is interesting because we know the benefit of like disturbance in a system, and we pulse the system all the time in order to create more light and more nutrients, and it's like they're considered parasitic, but also there's huge benefits for the entire ecosystem in the long run anyway.
And the honey mushroom is a really cool example as well. The honey mushroom is the biggest organism in the world. A lot of people might have heard of it. It's like a giant mycelial mat in Oregon in the States. The honey mushroom is typically a parasitic mushroom, but can also be a tertiary decomposer and saprotroph. So there's a really interesting example in Mycelium running of the honey mushroom basically causing huge blight to a whole lot of established trees and killing all of those trees. But the flip side of that is that it's opening up space for succession to start over again. Like Banya touched on in the succession class, like climax, old growth forest starts to taper off in its productivity. And so an organism like a honey mushroom in that climax environment where productivity is running low, things are getting shaded, trees are getting old, getting ready to die, the parasite might move in and create that disturbance, kill a lot of those trees. Primary decomposers will move in, start to break them down. Once competition arises from actinomyces and other bacteria, secondary decomposers will have an opportunity to move in. Those trees will end up falling on the ground, they'll break down a whole lot, and then tertiary decomposers can move in. So the honey mushroom is essentially making its own food source over a really long period. Their largest organism, I think, was about 1,500 to 2,000 years old, weighed over 50 tons, so it's a huge organism covering thousands of square kilometers, which is really cool to think about in that context.
2,400 acres?
What's that?
2,400 acres?
There you go. It's a big organism. Does anyone
Have any
any
questions?
questions?
Where is mold? This like primary, secondary.
Mold isn't really on here. Mold would fall under like decomposing. Oh, no. It would.
Tertiary?
Mold a fungi or a bacteria?
Yeah, fungi.
There's types of molds in both,
Yeah.
but mold is primarily fungi. They don't really I've never really read about it being classified.
It's like a class. Mold's like a class of its own.
But they are a decomposer, I guess.
Yeah,
It's
they're
not
part of
really the type of decomposer.
They're part of the fungi kingdom, though.
Yeah.
I'm just thinking if they're like a primary, if mold is like a primary, because it's like they're pretty much straight away, like if you leave something.
But mold feeds off of primary, secondary, and tertiary decomposers. So you find
But
you'll find
it's
mold.
like the first thing you see if you leave food somewhere. That's what mold.
I think that it's tertiary. What about?
Yeah, I'm pretty sure mold and algae are almost like classes
Their
of
own
their
little
own.
thing.
Yeah, they're gonna jump between all of it. Some molds will create like antibiotics and be beneficial, so they might fall into mutualist, might fall into primary decomposer for bacteria, primary, secondary, and tertiary decomposers for mushrooms. Sometimes they might even be parasitic. I think they're going to be all over the show.
I think there might be a visual in the sort of thing, it could be in Mycelium running, that puts these class of fungi we talked about and then some of the other branches of the fungal realm. I think there might be a picture that might put molds in relation to what we've been talking about with some of those. I think you might look at that.
Yeah.
Might be visual there to check out and look at for next week.
Yeah, lovely.
That shows that the branches of the fungal tree that might put molds in. So
I'd love to hear the answer from anyone who goes down that rabbit hole.
That was a bold. So, just
We did a quick search,
yeah.
and it sort of fits into producers. So they're kind of
They're
like
across the
you
whole
said,
board.
They're across the board. So you can't really classify them because different species thrive in common. And the same type of mold, which is what I was
Molds across the board.
So
Sometimes they're autotrophs, sometimes they're heterotrophs.
like you can get mold from sterilizing your substrate too hot, you get pink or black mold.
too clean.
Too clean. Too clean, it's moldy.
Might be trudging up.
Yeah.
Does anyone want to read out mycorrhizal fungi?
Mushroom related to roots, bring distant nutrients, moisture, and extends absorption zone well beyond the root structure of the host plant, and can also help to be a fungal defense against invasive diseases. We like it.
You like mycorrhizal? Cool. Well, we've got two types of mycorrhizal that we've got the definitions for as well, so maybe we can read them and see if there's any questions for it. Does anyone want to read out ectomycorrhizal?
Fungi form a sheath around the roots of partner plants, example chanterelles and Mycelium. Only 5 to 10% of terrestrial plant species have ectomycorrhizal and tend to form mutual symbiotic relationships with softwood plants, including birch, beech, willow, pine, oak, spruce, and fir.
And that we've got endomycorrhizal.
Fungi invade the interior of root cells. They are found in over 80% of plant species, including crops and greenhouse plants such as most vegetables, grasses, flowers, and fruit trees.
So cool little fun fact I heard about the ectomycorrhizal is in a forest environment there can be 10 to 100 times the surface area of mycelium than there can be leaves in the forest. So there's huge amounts of mycelium biomass in the forest, sometimes even more so than the organic matter itself. And that kind of ties back in, because fungi can spread so far, that comes back to that immobilization. So all of those nutrients that are being cycled and broken down from plant material from our primary producers is actually getting locked in to the biomass of the mycelium. So it's like something like 90 to 98% of all nutrients in the soil are immobilized. So there's not, besides conventional farming where they use liquid fertilizers and everything's just mineralized and ready to go, in nature things are getting locked into living organisms, which is really important to always come back to.
Because if it's not locked in and it rains heavily, we have, rain is a good example, like those nutrients will just be leached. But when they're immobilized and locked into the bodies of parts of the soil food web and into things like humus, then you have a much healthier and more resilient soil.
In terms of like conventional agriculture, it kind of comes back to that cation exchange capacity, which is like super important for how those nutrients are going to make their way into the plant. But in a healthy soil, like all those nutrients are going to be locked up unless, which we're about to get onto, unless a predator comes along and unlocks those nutrients. There's one more fungus to get through, is the endophytic fungi, which are one of my favorites. It's a big reason that we make compost teas and do foliar sprays. Just to put an idea of where they fit in. And does anyone want to read out the example?
They're found in all plants. They play a role in reducing herbivore stress, as well as support nutrient uptake, heat tolerance, plant evolution, and biodiversity. They live inside of plant tissue or in the roots, and a single plant can have hundreds of species.
I love some of the examples. You were talking the other day about herbivore stress and what we mean by that.
Has anyone heard about how like plants give off signals when they're getting eaten by a herbivore? So that's kind of where endophytes can fit in, because the way that they grow is they will get into plant tissue like through a leaf, and they will actually perform the same function. They'll replace that cell, but they'll be performing the same function as that cell inside of the leaf. So doing exactly what it did, except it can create antibiotics or toxins. Maybe wrote them in. They can, like all kinds of phytotoxins and all kinds of toxins, which will change the flavor of the leaf. So if that plant is getting eaten too heavily by a herbivore, like a cow, the endophyte can actually change the flavor or make the leaf poisonous to stop the herbivore from eating it, to give the plant a chance to grow back.
There's a bit in, I think it's The Secret Life of Trees as well, where if one tree's been eaten by a deer, the endophytes not only change the taste of the leaves in that tree, but the other trees around it will also be connected through the fungi, and they'll all change the taste of the leaves.
So they can give off like all kinds of pheromones and from the plant signaling any stresses. The endophytes will react, and they're like the defense system for the tree, which is really, really cool. Another example is like on an average day, there can be up to like 10,000 spores landing on any given leaf on any average day. And so if a lot of those spores landing on the leaf could be primary decomposers, or they could be parasites of any kind. If there's an endophyte living in that material, those spores landing on the leaf aren't going to have an opportunity to grow and potentially hurt the plant. Another really cool one with endophytes is when they're present on the plant's ready to put on flowers and seeds, those endophytes can actually be a part of that seed. So when you plant a plant, the endophytes might already be present and will just grow symbiotically with the plant.
So it's inherent in the plant.
Unless they're independently treated or sprayed.
When you said this is for our compost tea, when we do the fungal, you're trying to brew endophytes.
Yeah.
And how do you
Yeah, definitely. So there's also there's endo and ectophilic fungi as well. Some, not all live in the leaves. Some will live in the root zone.
Endophilic would be the parasitic
Inside.
bit.
No, not
Sorry.
all the time. Though they can still be a part of the defense of the plant. But yeah, none of these boxes, nothing's set into these boxes. They can definitely be parasitic in the box.
So it could be, you don't know what strain you've got. It could be parasitic strain.
I wonder if before we move on to the next step in the soil food web, whether we can just recap how the relationship between plants and their exudates and bacteria and fungi in terms of soil chemistry and I guess like mining the soil for that exchange maybe might be useful to kind of recap this part and then how it then gets locked up and then we'll cycle back around.
That example we had at the bottom.
Kind of. But I guess just recapping about, I know that there's a lot of people that are fungi and understand their role in plant health or in terms of becoming the extension of the roots in order to bring nutrients back to the plant. And I thought, yeah, maybe we should just go back and talk about that relationship before we talk about then the things that eat that first trophic level, second trophic level. Not clear?
Like,
yeah, go.
Plants need minerals and nutrients to grow. So to get them, they produce exudates, sending out that secretion in the soil. Then that signals the bacteria for that symbiotic relationship to be formed. The fungi are more of the mobile ones, so they might go far out in the soil to bring those nutrients back into the rhizosphere of the plant. And then the bacteria, the fungi, are like your little fertilizer bags. They ingest the minerals that have been brought back into that rhizosphere. And then when the plant needs them, that's the bioavailable form. What is it? Ingested by the bacteria and the fungi. And then when that decomposes, then the plant can uptake that nutrient and grow.
yeah, I think you're crushing it. I think the fertilizer bags is a perfect way to explain bacteria and fungi, like they're locking up, immobilizing everything in the soil. When they die, it doesn't necessarily release all of those minerals. A lot of them might stay in that structure of the bacteria and just be locked inside of it. It's not until we get to kind of the next trophic level where we start moving into our nematodes being root feeders, fungal feeders, or bacterial feeders and predators are the guys who come and eat the bacteria and fungi, and their excrement is what's bioavailable to plants.
That makes sense to me. So yeah, using that tinning with microbes analogy, that these guys are the bags of fertilizer. The next layer are then what we'd call fertilizer spreaders. So they eat those bags of fertilizer, excrete them out in a form that plants can then uptake. And not just plants, but other things further along that trophic level. That makes sense? Yeah.
So there's I thought there was like a direct exchange between the mycorrhizae and the rhizosphere.
Yeah.
And the things, yeah. Is that what you're saying? Like, I didn't, so were you saying that the fungi and bacteria would need to be eaten, decomposed by the next level before the plants could uptake their...
No, that's a good point.
Yeah.
Mycorrhizal would be the exception to that because they're linked to the plant, whether it's inside or right on the outside of them, and in exchange for those exudates, they are feeding the plant. So it's in not all cases they need to be eaten by a predator to release those.
There is like a direct sort of transfer.
like bacteria and fungi differ slightly in how they make the nutrients more bioavailable. Like fungi release enzymes and acids to break down these molecules into simpler forms, releasing the macro and micronutrients to then be uptaken. Whereas bacteria, correct me if I'm wrong, if they eat them and then excrete them as like poop, or do they like shred them, or is it then decomposed?
Yeah, so there's predators and shredders. Predators are eating that bacteria, which is, and their excrement's going to have soluble nutrients in it. Cool.
So basically, that there's like a few different ways that the nutrients in the soil get into the plants, but it's all through ongoing bacteria somehow. Or is that also like there's also the exchange of like water being uptaken and chemicals in the water and stuff?
Yeah, so and we are trying to avoid chemistry, but not all minerals are like mined. Well, no, pretty much all minerals can be mined by bacteria or fungi, but a lot of them get broken down through weathering or through like geological events, and so they typically are really prone to leaching. So a lot of nutrients that plants are going to get will be carried because of the increase in surface area of like your mycorrhizal. Would be more typical that the mycorrhizal are going to catch a lot of those nutrients that are being leached out of the waterways.
Is everyone still with us?
I reckon we smash out. We've got a couple more guys to finish the soil food web, and it might make sense to have a break.
Yeah.
Cool. So next, after fungi, we've got Protozoa. Does anyone want to read out the definition of Protozoa?
Yeah. Protozoa are single-celled predators which feed on primary decomposers, mainly grazing on soil bacteria around the root zone, releasing excess nitrogen and nutrients locked in the body of the bacteria.
And then we've got a few different types of Protozoa, which is more in relation to their size and shape. These are going to be some of the main guys that we'll be able to see under the microscope when we get to that part of the class. So we've got Amoebas, Flagellates, and Ciliates. And does anyone want to read out what an amoeba might look like when we have a look at it?
They can just keep changing.
They're like blobs. Some of them have a protective shell, and some of them don't. So when we look at them, we might see little circles. That will be them hiding inside of their protective shell, because when we make a slide, they're going to all get disturbed, and a lot of them will slip back inside of their little protective home. Flagellates.
Yeah.
Like a sperm?
Yeah.
What a sperm looks like. They're your flagellates. And the flagellums, which are the tail parts, can be on the front, the back, the sides. We're mostly just looking for the flagellum to identify a flagellate. And then we've got ciliates. Lots of little hairs on them, and they use them to swim around in the environment. Ciliates are kind of the main predators in the protozoa world. They'll be eating a lot of the flagellates and amoebas. All of these guys typically are going to be found in the rhizosphere, where there's the most activity from the exudates from the plant. I put my money on us being able to see at least two, if not all of them, under the microscope later. We can talk about them more.
Blastocysts Sominus is also a protozoa, right?
That'd probably be a Google question.
That bacteria.
It's like a common parasite around here.
Alright, last one.
Just on
Yeah.
that, to have considerable healthy soil, you need a diversity of all three.
I think you'd need a healthy diversity of amoeba and flagellates, and if you did have a healthy diversity of them, then you would end up with ciliates being in the environment to feed on them. So, yeah, hitting protozoa, they're our next trophic level after what we've just been talking about, and they're predominantly grazers and predators. So, think yeah, without the grazers or the predators eating our fungi and bacteria in that rhizosphere, all those nutrients would stay immobilized. So having them feeding off the super high diversity of bacteria and fungi in that rhizosphere, these guys are what coming through, eating them, and then having those nutrients available for the plant to uptake. If they weren't in there eating bacteria and fungi, those nutrients would stay locked up in the cells of those organisms until they eventually get eaten by a predator.
Wait. Protozoa.
It's protozoa. Nematodes are also predators, and they're kind of spread throughout the trophic levels, which is a lot because of their size, but also because of their function. And their presence will be dictated by the succession as well of what plants are around. Does anyone want to read the definition for nematodes?
Nematodes are any of the phylum Nematoda of elongated cylindrical worms parasitic in animals or plants or free-living in soil or water. Nematodes feed on living material, making nutrients locked up in living material available to plants and microbes. They're considered grazers. And fun fact, apparently if you took away everything else on Earth apart from nematodes, you would still be able to see the Earth from space because there's that many nematodes like in the soil, in our bodies, and everywhere. It'd just be like a silvery orb of worms.
What did you say?
For every one human, there's about 60 billion nematodes.
Yeah.
And on your average square meter of soil, there's 1 to 10 million. There's a lot of them. And they're like really, really primary at releasing nutrients and mineralizing nutrients through their excretions. Another cool thing about their excretions, it's really similar to worm castings in the sense that it's a really stable form of organic matter that's coming out of from their excretions, and it's 40 to 60% of the carbon that comes out of them is in a stable form and can be sequestered, as opposed to your bacteria, which through respiration, through exhaling, they're respirating carbon dioxide, and they'll respirate about 2/3 of the carbon that the plant stored in its organic matter. There's lots of very wordy, but
And the nematodes are already much bigger than bacteria, like bigger, like 100 times bigger or something.
and the size. Like your root feeders are your smallest, your omnivores, bacterial and fungal, slightly bigger, and your predators are your biggest. In terms of nanometers, I can't remember off the top of my head, but hopefully we'll get a look at some of them.
How much did you say that the bacteria release?
Through respiration, 2/3 of the carbon. Which is why our compost piles, we start with a big pile and end up with a small pile.
And just before we go on a break, I feel like the definitions of different nematodes we've kind of covered, even if we don't think about word for word. just to sort of touch back on that soil food web stool before we have a break, looking at the soil chemistry and the unlocking of those nutrients. Like, are we starting to see how the soil biology is relating to the soil chemistry at a very microscopic level? And then also how the biology is also creating this structure, and it's the movement of the nematodes and then larger parts of the soil food web that is creating that structure as they move through. And especially, we're talking about in the rhizosphere, moving through that rhizosphere is putting in oxygen, it's depositing nutrients right in that zone, eating the bacteria that's full of the little fertilizer bags, and then depositing it right in the zone that the plant can then make it accessible. So we might take a break. Just give people's brains a little break. And we'll, maybe like 10 minutes. Come back. Yeah.
Last 30 here.
Stretch.
Yeah. There's a little bit of if you want to look through it in the break, there's a little bit of an example of what Greta was just talking about at the bottom here of how example of soil food web and macronutrient cycling. Just to tie a few of them together on how a lot of those macronutrients might become available to plants.
So, this is a class that we did a couple weeks ago on ecological succession. So, I'm going to let these guys work through succession as an overview for you. 2, 3, 4, 5, 6. Come on, trees. What are you looking for? Perennials. That's yours. Cool. So, in the last class, we kind of ordered what we thought from bare soil to old growth forest. What order, like the succession would go in. So that would be like the last one, part of the trees and like a complex sort of forest system. And if you were starting with bare soil,
how do you get it? What needs to happen to get into a forest?
Yeah, we did this, and now I think we're gonna think about how all these aspects like link into this.
Can we ride on it? Like on the back?
Look at Greta's face. I was like,
They are really nice pieces.
Okay.
Start with the bare rock.
So yeah, start from the very beginning. Bare rock and soil.
That's where it's all happening. It's all going
That's
on.
where it's all at.
Yeah. I reckon there's a lot of bacteria.
I would say if it's bare rock, there's a lot of fungi action going on as well.
There definitely would be both, but it would be more like things like lichen. If there's, yeah, perhaps that's like at this stage it's like isn't that like 1,000 to 1 bacteria to fungi. So there's like a little bit, and then getting more into that here.
Sunday each one you can test them.
One interesting thing regarding lichen, if you ever read a book called Red Mars, Green Mars, and Blue Mars, Red Mars is the first thing they do is spray lichen all over Mars because they're trying to create an oxygen environment. So the lichen can function in an oxygen-free area, and then they start off-gassing oxygen and then they warm the environment, so you start to create an atmosphere in the environment.
So lichen, old man's beard that hangs off the trees, or Jupiter's beard, or
It's actually bromelia, I think.
what? It's not quite a fungi.
Bromelia, I think.
Really? I think so. I've got some in my bag.
Nice. It's not quite a fungi or a bacteria because they both live symbiotically to work together. Like they need each other to live, so it's kind of like something in the middle, which is where they fit into here nicely because there would be so much bacteria, and they would start to cover the rocks and digest them to bring more minerals into the soil.
Anyone?
What you said then is a really good example of that mineralization. So taking something like a rock and turning it into a veil.
You also see lichen all the way up in the Andes to the top. And then, is maca a lichen? Maca, the maca root.
I think it's a plant. I don't know enough about that. I'm not sure.
In other words, back to what you're saying, it would be immobilized inside the rock.
Yeah.
So that has that.
It consumes it.
Yeah.
They're actually breaking rock now.
Should we? I feel like it'd be cool to have a look at some of the words we've learned as well and see where we can fit them in. Yeah. So between when we've got the lichen, mosses and fungi, going from that into the next stage, like grasses, annuals and weeds. What other ideas have you got for that process?
Question, so when we talk about the first grasses, we're talking about stuff like farmers' friends?
Yeah.
But, so the farmer friends basically are bringing nitrogen into the soil, cracking and opening the soil up?
They're not a nitrogen fixer,
No?
but they would be putting out exudates and feeding the soil and helping to open up. I'm sure they probably have some specific nutrients that they would mine, but I'm not sure what they would be.
So having farmers' friends in our fields is advantageous? Or does it mean the soil is not that healthy?
At that stage, going from there to there, yeah, farmers' friends are great, but it can also start to interfere with other perennial grasses, which are slower, and shrubs. You'd hope that through the succession, you would start to end up there, but it can, because of its aggressive nature, it can dominate.
That's why I've been watching it. I'm just looking at what we see here, where we see them in the soil, and we were saying they're advantageous because they're making up the soil, but we're hoping that eventually they go away and the aggression will take over.
Yeah.
I think in the first stages of succession, anything that's covering bare soil is initially is advantageous, just to get that, improve the soil environment in general and the soil health. But eventually, we'd want it to be replaced with things that we actually want further down the line.
So between the breaking down the lichens, moss, and fungi to grasses, annuals, and weeds, which all of those things are the autotrophs.
Yeah.
So we're somehow moving from
autotrophs to heterotrophs.
Where do you think heterotrophs start to appear?
So the autotrophs, what they're doing is actually turning minerals into nutrients, or they're making nutrients available to be uptaken by the next trophic level, right? So the immobilization? Yeah. Is that right?
Yeah. Yeah, because that would be that organic materials.
Yeah.
So if now there's these nutrients now available, then we're going to be able to move to the next layer of or next trophic level of the food web, which should be things like Protozoa.
What's on that second to last?
Small
trees,
kind of
small trees and softwood species.
So if we're here and there's a mobilization of minerals.
Hardwood trees,
grasses,
forest systems, and climate.
Annuals could be moving towards maybe,
or with the rhizosphere, the annuals, there'd start to be some higher levels in this food web.
Yeah.
Not higher, but more down the web.
So more like Protozoa.
And the nematodes start coming in, right?
As you start to get that larger bacterial and particularly bacterial in here, and then as you start to go to larger stuff, more fungal dominated, then you start to have the things that will predate them.
Nematodes, we got root feeders. Root feeders. Probably more bacterial feeders and root feeders.
And then them coming in. Now, I was wondering how that would impact like the succession between grasses and pioneer species and stuff.
Well, bacterial and root protozoas, their exudates would unlock another layer again.
Yeah, and they build the soil health as well, like the exudates cause aggregation, because aggregates form when secretions clump.
Yeah. Which would, if the soil conditions are better, then any seeds or things that have flown in or being carried by the wind or already dormant in the soil now have an opportunity to grow and utilize that space that's been created. So I think maybe as we move into pioneer species and nitrogen fixers, we might start to get a more broad range of protozoa, like maybe then the predators and maybe some fungal feeders.
Yeah, we're slowly moving into more fungal dominated.
I feel like, it's fungal. Yeah.
Definitely like in the old growth at the end here, it's very fungal dominated.
Yeah.
But they'll still
Beginning
be like,
it's still.
yeah. But it's weird.
In terms of Protozoa at this stage of succession, it would be more Amoebas and Flagellates.
Yeah, because then the amoebas and flagellates lead to better ciliates, right? But when do you think that would come in?
Probably when soil health improves and soil structure becomes more more at this stage.
There's greater root structures with this one, so there is going to be more that speaks to the structure of the soil overall, and it's just going to keep improving. So do you think amoebas and flagellates start to evolve, and then maybe all three start to come in over here, or maybe further along?
Oh, that's sure.
You guys are trying to map out protozoa.
Yeah, thinking about like, because Cal was saying how like amoebas and flagellates would kind of come first, and then the ciliates would.
Once there's a food source for them.
Once there's a food source for them. So, like in terms of the succession, Cal was saying he reckons like when the soils become a bit better, like around up here, that's when the ciliates.
Weird. I think that's pretty fair. Like, yeah, I don't know. I've talked to Michaela a whole lot about it. Like, in terms of this exercise, it's really cool to start thinking how things kind of pass the torch along and evolve. To be, like, there's going to be the whole diversity of the whole soil food web, like, even at bare soil or right at these beginning stages. But so we're trying to map out when things are, like, dominating or beginning to affect succession. So I think that's a great way of putting it, is after the amoeba and flagellates have a really healthy population, there's going to be a more abundant food source facility, which I think is perfect.
And then after like that's with the nematodes, because we were saying like nematodes would still be present, like even with like.
Yeah, well, as soon as plants are around, root feeders are going to show up for sure.
Root bacteria.
But in terms of like impacting succession, maybe they're not going to have a huge impact until the nitrogen, like woody legumes and stuff, start to show up, and they're going to go to town on those nodules to get that nitrogen, and they're going to have a huge boom in population, and they're going to affect succession that way.
And then
There's no right or wrong answers, and this is just
When there is the available space and food, how do they reproduce? Protozoa and nematodes.
Nematodes mostly laying eggs, and protozoa, a lot of them do the two cells split apart.
Division.
Yeah, type of division. I can't remember the technical word.
Mitosis.
And they'll only do that if it's the right environment.
Yeah, well, once they've got enough food source to.
You know how bacteria can, or is that viruses that can like share their chromosomes? Is it like chromosomes or like their DNA cross to each other? They can like inject DNA into each other. So one will take on. Like,
like a virus.
Like yeah.
Like the circle thing.
Viruses are RNA.
Those ones. Right? A virus? Like it's got a ring structure and you can splice. Fungi. So we've got lichen being kind of like a cross between fungi and a bacteria over here. And what do you guys think? Where do you guys think primary, secondary, and tertiary would fit in on this gradient?
Predominantly. Or the action under the soil.
At the beginning?
Yeah.
Because other things like bacteria, like the bacteria are already creating that partially digested material. That, like, for example, a secondary decomposer could produce for that. So yeah, they're more over here.
Secondary primary would be like up here, breaking down the.
So I reckon you'd have a lot, where you've got bare soil, you'd have a lot of your autotrophs, wouldn't you, to begin with?
Yeah,
things
true.
that can that can make their own nutrient.
So like.
And also these lithotrophic bacteria, they produce their own nutrients through oxidation of inorganic compounds. So you'd have a lot of those. They would be quite a dominant life form, wouldn't they, before there's much else going on, maybe?
Yeah, that makes sense. The only thing I wonder about that is like how, because bare soil, when I think of bare soil I just think of like a really unhealthy dead environment. So I'm like, where?
No, because all of this stuff, if it's a living soil, it's got all of this stuff going on. But I think what Brian was saying is from a successional point of view, is that by the time you get down here, you've got the whole orchestra playing of all of these things, whereas at the beginning, there's just a few sections of that are louder than others. So, you know, you'd still have your lithotrophic bacteria all the way through here, but they've got more of a role here compared to everything else that's going on, rather than your climax where you've got fungus and all of the different trophic levels, all of the different ecological things, your consumers, predators, everything's going on at the end there.
Yeah, so in terms of like the first ones that are there, like the autotrophs and the lithotrophic bacteria, right at the beginning, I feel like, it must, the fields that are just like open and clouds and like dry and just like...
It's kind of what I have in my head.
Yeah, but I guess they're in there in some quantities, and then when you put on a cheeky bit of mulch, it might improve a little bit, and then a little bit.
Yeah, you've got all of them throughout all of here, but if you think of because they're different ecosystems, so you've got different things playing more of a role in different ecosystems. Same as if you're just looking at the plants. So you're saying that they're like predominantly these are the sorts of plants that are in those
different time frame within an ecosystem.
Yeah, but with the soil life, they're all there all the time.
Yeah. So you're saying that even in like bare soil, you would have like all of the fungi and everything, or are you more saying like you'd have it all up here, but down there? Just...
You could have some fungi here, but they maybe would be fungus that's living on bacteria. If there's such a thing, is there fungus that eat bacteria? Penicillin does.
Yeah, there would be, like, fungi can dominate over bacteria. I guess I've only seen bacteria being a problem for the fungi, but
if we're thinking about like the very start of succession, I feel like that's already with succession in mind. So like there'd be trophic levels there, like fungi feeding on bacteria. That's like a trophic level. But like if we're taking it right back to the beginning, of, like the first trophic levels, was it the lithotrophic and the autotroph you were saying? So they'd be like dominating right there.
So I guess it's when in this whole scheme do all of these fit in, like start to dominate, or start to become more prolific, because they're always going to all exist throughout the whole time.
And depending on the context, from the start is what's going to be present already.
Yeah.
And I guess all our concepts or context.
And depending on the climate, it's going to change how quickly they all evolve.
And then, so after the lithotrophic autotrophs are dominating in the first place, then we've got like the lichen.
So we've got more mutualistic bacteria.
I just get so like caught up on the whole bare soil thing because I'm like in the beginning when there was.
But you were talking about evolutionary time, weren't you?
Yeah.
Whereas, you know, we're not there now.
I know, yeah. But it's interesting that when you think about rock, like right at the start, because it was just completely a fungi world before, and then it became bacterial and fungi. I think that's my.
But I suppose if you started off with a completely sterile environment and you were starting your own little creation world, that would play out. But there's everything all interconnected already.
When do we think pathogenic bacteria would come in? It causes diseases in plant cell walls. So
I guess as soon as there's plants. What else we got in there?
Okay.
Do you think the pathogenic bacteria ever really dominate though? Like I guess if there was a real disease going on.
Particularly if they're anaerobic.
Yeah.
Really heavy waterlogged soils, like after a big flood or rain.
which would happen when you've got shit soils.
Yeah, I think so. But yeah, even just like really like floods, a meter of rain. Yeah, they definitely could. But I feel like in an anaerobic environment, I'm sure there would be a limiting factor within that soil food web to stop pathogenic bacteria from becoming the dominant form of bacteria in the soil. It's not really going to be beneficial for the rest of the environment. I don't know what the age would be.
Like endophytes might help too. So if a pathogenic bacteria started to dominate, then endophytes they'd come out and have war. So that's like that's a wormhole for education. Like if this was to happen, then this would happen. So we all agree that for protozoa, you're going to start off with amoeba and flagellates, and then as they have more food source, for ciliates will start to predominate. Well, now, what do ciliates need exactly?
Amoeba, they eat the amoeba and the flagellates. Okay. So when they've done their blooms, they have enough food.
So the same thing would kind of apply for the nematodes. You've got bacterial and root feeders, and then the predators would come when they've got stuff to eat.
I think you would still find amounts of them here, but maybe just not in as high amounts.
Yeah, yeah.
Would you wouldn't find worms in like big worms, loads of worms in like really unhealthy, dry, like barren sort of thing. But do you still? No, yeah. They come from. They just come when it gets healthy.
Like where they come from?
Like animals are the main movers. Like me and Brian were talking about, like how you get bacteria and fungi into a place, and it's all just mainly through animals, and especially things like worms. Like if you get, like you said, the eggs, worm eggs, they'll get stuck on animals' fur and their feet as they move around. And also, like, I'm sure worms can travel a pretty long distance.
Yeah. And do the worm eggs survive in like a bird's tummy?
I don't know. I don't think they would. I can't imagine they would. Maybe they would, but probably not. Yeah, through water.
There's so many different kinds of worms as well. Like the kind of worms you'd find in a bird's
different
tummy
than
are
how we
probably
think about
parasitic
what
worms
might
on the bird. But if you're talking about worms for the soil, they'd be in the soil waiting for the organic matter. And
but would they be in there waiting, like, or would they come?
We're going to all go debrief in front of the microscope.
I was confused about
Well,
the
yeah, but I think that all you need to like start to have worms growing is like one egg.
Yeah, like one pregnant one.
One egg, one pregnant worm.
yeah, and they're so tiny.
Yeah.
Like Ivan said, they can get carried on our shoes, on birds' feet.
Yeah.
One egg.
Or feathers.
It's like if you build it, they will come. That kind of yeah.
And one egg can have like 15 worms.
Yeah. That's so crazy. Amazing how.
100 times magnification of some compost that Cal picked up just before this workshop.
It had like massive strands of mycelium.
And we'll zoom out and take. I've already seen some hype there, and they're all.
Holy shit!
That's a nematode moving around. Should we get a better look at them?
Guys, like Nat
Relax.
Geo. It's a flagellate. It's live.
He wants to go away.
Where's my?
It's like being on safari, you have to be very quiet.
Okay.
Be scared.
What's the big black blob?
So that's probably.
Oh.
Go a little bit more up, there's something.
Potentially a piece of hyphae over here to the right. That big black blob will just be a piece of organic matter. A lot of the smaller particles might be sandstone clay, might be dead pieces of shredded bacteria, or larger. Where did that go?
We'll go down. There was something moving if you go left and then down a bit.
So can you see protozoa, or they're too small?
Yeah, at the last magnification when that thing probably
Something!
So that'll be a ciliate running around.
That's a protozoa.
Yeah, so that's a protozoa. So he'll be cruising around looking for food. They move really quickly with those little ears, especially in a water environment. All of these slides, I've added water to them, shaken them up, and put one drop of each on here. So this is just one drop of compost liquid.
And before, when it was zoomed in, those things moving around were bacteria.
Yeah, so the reason we went through all these different kinds is when it comes to actually identifying what they are under the microscope is really difficult. We're able to see nematodes, but we won't know without being able to see its mouth parts whether it's a fungal, bacterial, omnivore, or predator. The size of it, we might be able to say it's a predator, but the way they do that is by freezing the slide, and then they can zoom right in and actually identify the mouth parts of the nematodes. And our amoebas, ciliates, and flagellates, by the way they move, as we just saw that.
Oh my
Because I've got this light underneath the slide here, he's going to be getting like warmed up from that, and he's probably not very happy about it, which is why he's just going to keep moving around.
Oh, now he got it.
Wow. And so a lot of that stuff that's bouncing off his body, that's
That'll
like
be,
doing
yeah.
so, and then it gets like, oh, he's eating.
The correct. He's not having a good time because he's getting cooked right now.
What kind of
But I mean, he usually lives in a hot compost. Doesn't he live in a hot compost?
So I'm pretty sure Cal got this sample from the compost pile up on the hoop. Yeah. So that's a few months old now and has been resting for a really long time, which is great to see our predators starting to move in, which is going to be forming a whole lot of soluble nutrients when we use that compost in the garden.
So if it was like the hot compost, if it was the hot compost, do you think that we wouldn't see like guys in it?
No, yeah, if it was hot compost,
Yeah.
would be
You killed him!
would be really So nematodes, they can be as deep as I read out of Mycelium running, I think. No, I can't remember where I got it. It was 0.9 to 3.6 kilometers deep. These nematodes can live. 90% of
0.9
them,
to 3.6 kilometers deep.
but 90% of them are in the top 15 centimeters of your soil, which is why tilling can have such a massive impact on your nutrient availability in the soil and its ability to cycle nutrients. Because that's your main area that you're going to be tilling. And in terms of them coming back to life, farmers have a huge issue with root-feeding nematodes. They till their soil, plant their crops, and they all get eaten by root-feeding nematodes because they're the earliest ones to come back in that succession. And they'll smash all the plants, farmers spray pesticides. So that is really good looking. A beneficial, that's a hyphae. So that's a piece of mycelium. And typically a beneficial mycelium is anything over 7 microns, so 7 bacteria stacked on top of each other. Anything bigger than that size or bigger, is typically a beneficial hyphae. If it's white or like see-through, it's really likely it will be a pathogenic fungi. This one having a blue tinge to it is a really, really good sign. It's probably a really beneficial fungus.
How do you know that that's
Just by the uniformity in its shape. We can try and zoom in and you see little nodes that run down it.
So that nematode is eating Protozoa.
No, so it'll either be, there's 5 kinds of nematodes. It'll either be a root feeder, fungal feeder, bacterial feeder, omnivore, which will either eat bacteria, fungi, or sometimes other nematodes, and a predator nematode, which will only eat other nematodes. So to keep you till your soil, plant your annuals, they get demolished by root feeders. The root feeders, fungal feeders, bacterial feeders, and omnivores need to show up before there's a food source for the predators to come and keep those root feeders in check. So it can take a really long time for your soil to recover from being tilled.
fungi, bacteria, root, omnivores, and
Fruits.
It's good.
it's actually and so we've got compost, and what else? What other samples have we got?
To the right.
So this is back to 400 times zoom. See if we can. Might be able to make this up on the screen.
oh yeah,
Yeah, we can see it. Have a look.
It's nice to know.
everywhere.
Let's see. We're going to go back out to 40 times zoom, I think. We're just going to have a bit more of a bird's eye view. Everything. There's our nematode again.
What should we call it?
Your name.
Nemo.
So we can. I've got three.
John did a new game on the weekend.
There's
Something here on the left.
another really large hyphae.
Is that a pathogenic?
It does look pretty clear, but this is out at 40 times magnification, so it's very large. So it could go either way. We're going to move over to the worm casting.
We could also have our lunch in front of the TV. Yeah. If people feel like they want to have TV dinner.
There's a little hyphae. This is our worm casting. Lots of really good looking aggregates, all these little parts, soil particles that are going to be created by the worms, which is going to be our stable organic matter that's going to last a really long time. Once we put that out into the garden, it's going to have a lot of available nutrients in it. Nothing too exciting, just jumping out 40 times. Might be some like that.
That's the worm casting.
This is worm casting, yeah. That's a crack on our house. Yeah, really good looking aggregates. You can see it's almost like a cloud in there, which is just so that's been taken down to almost like the clay consistency, and it's going to be mostly just available nutrients in that kind of haze that's in the back of the stream, which is awesome to see, and that's what we'd expect to see in worm casting.
But herpes bacteria only affects, there's different strains of
All
herpes
wiggling,
bacteria.
yeah.
And it's the ones that didn't.
Oh my god.
This is our worm casting back at 400 times magnification, and everything that's moving in there is alive.
This is the maximum.
As much goes up to 1,000 times.
Oh, I can see something moving.
But that only works in
Everything is
times.
all, if you go
I
close,
don't
they're
know
all
what to-
moving.
Colours.
That's your eyes, Sarah.
Is that all live bacteria moving around?
Yeah.
Whoa.
Oh,
party.
you guys should come up close.
We can all see.
actually identify. Typically they're identified by their shape, so you'll get like rods or spirals or like multiple rods put together is usually how they're classified. So when you get a biology test, sometimes they'll classify them into the shape of it, which you can go back and get a broader idea of what they might be. But usually they just do a count. They'll dilute this more than what this is. This is like concentrate, diluted 5 to 1. They'll keep diluting it until there's not many bacteria on there. They get to a point where they can literally go to a screen like this, count how many bacteria there are, do that randomly 20 times, total that number up, give an average of it, and that's how much bacteria you have in the soil. They'll do the same for Protozoa, Nematodes, and
Yes, exactly.
Tide.
Yes.
it is a
Well, they're
tide. So
running
because
away
this is
from
only
something.
one drop, and this light is warm, and in maybe 10 minutes time, this will just, all that water will be gone, and so it's literally, it's a tide, basically. It's warming up.
But what we're looking at there, we can't see bacteria.
The majority of like the things moving in one direction will be particles, but the things that are more jiggling around in there, they're all bacteria.
I think is those little yellow ones are the things. Are they the Ciliates? The little frogs?
That one zoomy one that we saw going around was a Ciliate. See how our compost looks.
So you got them all on there.
Composting.
Ooh. Is this the fungal or the bacterial?
Oh, it's a circle.
Okay, I'm just like, how I see something. It's like I look away for one second.
Is that an algae? A blob of algae? Like a single
I think that's just a little air pocket. Sometimes you'll see, when you see really perfect little circles.
Oh,
Yeah. So this is at 100 times. This is at 100 times.
Yeah, exactly.
Oh yeah.
Go right.
They look like
Yeah,
a
cool.
protozoa.
God, it was crazy. That's so great!
It's eating a stick.
Oh,
I see it. Can you see that?
Yeah.
I ate something.
So this is great to be seeing this many ciliates in our compost tea.
What's that thing on the bottom left?
We should go closer.
Oh!
What was that?
It's a dangerous world for those ones.
It was like an explosion.
Like an earthquake. What's that? What's that thingy? The screen.
Whoa! Wow, that's moving fast.
We can try to
We'll just go to 40 times and see if anything jumps out of this. But yeah, to see that many predators in our compost tea, we're going to get, that's going to be unlocking and remineralizing all of our organic matter back into plant available nutrients. So in terms of putting that in the market garden, it's going to be perfect.
When you spray compost tea on the leaves, do they die, the bacteria? If they were to dry, would it need for that to actually drip down onto the soil? Yeah.
Yeah.
So would it be better to spray compost tea during an overcast day or early in the morning?
Do it in the afternoon. And there's also this with spraying at compost standards, Soil Food Web recommends that you only spray it out at low pressure, like 20 psi. But we kind of can't do that because we have too much to get our sprinklers to run. We have to run it a little bit more, so we probably lose a little bit of life through that high pressure for sprinklers.
How can they recommend the low pressure? Is that
Because like the force when
it
it's
can hurt?
getting pushed out, it can kill. It does kill bacteria and nematodes and things. It's like getting shot out of a big cannon.
Go do your job.
Yeah. Yeah. Exactly.
Now we see, yeah.
Now I see this. I really.
This is back to 400 times zoom. Yeah. What Ivan said about the 20 psi is really important, and then UV is going to kill them too. Spray in the morning or the evening, preferably the evening most of the time. A lot of them aren't going to survive on those leaves, and they can solubilize. Like if there's protozoa going out in the leaves, and there's other debris that's blown into that, other bacteria living in there. You can see how much is in one drop. If that's on the leaf, those protozoa can make those nutrients available, and the plant can absorb them through its leaves as well. So,
Really? And the cold? Would the cold kill them? If we sprayed it and it got very cold?
temperature's going to have an effect on them. There's different degrees of temperatures that some organisms like when the compost pile gets over 45 degrees, you move into like thermophilic bacteria, which enjoy that temperature. A lot of them live in the world of archaea, and some of them are classified as like your extremophiles and things like that. Yeah. Lots of different diversities of sharp.
But I feel like them tarping beds must have a massive effect on the bacteria life.
We tested it. We did microscope analyses of soil from under the tarp, and because I had the same thought, but they were fine. Like it does get hot under there, but if you take that tarp off, like it might, like the top layer might dry out, but like really talking about like that much soil.
But
and, like,
I'm just thinking it's both like the temperature, but it's also like it must be like less oxygen, but like available.
I think you still get a lot of air coming in through the edges of the tarps. It's not like we completely seal it. I think you would still be getting enough.
And they like the moisture.
Yeah, and it doesn't get in summer. It does get potentially too hot, which is why we sometimes run drip line underneath them. But this time of year, it's fine.
What were you saying about the soil? Like, say the top layer might dry out, but did you test
underneath there, like it's going to be wet. Like it's not all.
Yeah.
Just because like that top layer is dried out or it's hot. Yeah.
Whoa.
Like the temperature holding capacity of the soil is quite high. So just because that top layer might be warm and dry doesn't mean the rest of it will be warm and dry. But yeah, I've had the same thought.
I just feel like tarping just seems like such a weird thing.
It's better than that soil.
Yeah,
Things from UV.
Yeah, it's better than tilling.
Some of the things should be used sometimes.
Maybe you should try the Byron show.
What would you use?
Oh yeah.
I have no idea. I just really don't like tarping.
Oh, yeah.
I fucking hate it.
Yeah.
Yeah, you put biochar over?
They wouldn't last.
No, but there's lots of photos available.
And you're using it to kill and crop off instead of tilling. Like you're using plastic instead of using tractors, and I think that's. The guy has done the same thing, but it's so much better for the soil. Like, if we can use tractors instead of using a rotary hose or a
Brian, what's this kind of application?
This is 400. Which this is about as far as we can go in.
Is that your microscope?
We got it from the guy called. This looks like a cyst up here. And a little clear bubble right here. That might be a testate amoeba hiding inside of that cyst, protecting itself.
What is he hiding?
Just protecting itself. So when I shook up this slide before I put it on, it would have gone and protected itself. from a guy called Chris Ellery who teaches